We're very excited to have this upcoming Congress on lymphoma and development, and specifically talking about what are the new agents in T-cell lymphoma. I think it's a very exciting landscape, and there are a growing list of novel agents that are being in active clinical development for patients with peripheral T-cell lymphoma. And, I think I want to start by having some background discussion for the T-cell lymphoma, why it's so important to have novel agents and many different targeting novel agents, because T-cell lymphoma are heterogeneous and quite uncommon. We have over 30 different subtypes, and only about 5-10% of all non-Hodgkin's lymphoma. So it's quite complex, right, to make the best treatment recommendation because there's so much variety, complexity, in terms of subtypes based on cell of origin and driver mutations, as well as clinical presentations. So I think, therefore, understanding the biology and pathogenesis and potentially targets behind each of those subtypes, it really helps us to design, the rational and optimal therapy. And so we do have a growing list of novel agents in T-cell lymphoma. And I want to start by discussing about this anti-CD30 target agent, which is called Brentuximab vedotin. CD30 is abundantly expressed in subtypes of T-cell lymphoma, such as anaplastic large cell lymphoma, and perhaps to a lesser extent in other type of PTCL subtypes. And therefore, it's important target, Brentuximab vedotin. The addition of that to chemotherapy backbone, such as CHOP, has really made a significant beneficial impact for treatment of T-cell lymphoma. And it has improved survival, including progression-free survival and overall survival in the frontline setting, those patients with CD30 positive peripheral T-cell lymphoma. And that has led to practice changing a breakthrough FDA approval for this agent targeting CD30 in the first line setting. And there are many other agents and targeting different therapeutic targets in T-cell lymphoma. Some are already approved for use in T-cell lymphoma setting, but in the relapse refractory conditions, others are under active clinical development. And I can give you some examples of that. One of the important therapeutic arena in T-cell lymphoma is epigenetic-targeting. Genes that regulate, histone deacetylation or DNA methylation, histone methylation. They're very important in terms of pathogenesis in T-cell lymphoma. They occur quite commonly in subtypes of peripheral T-cell lymphoma, and therefore therapeutic agents targeting epigenetics are in wide clinical use. The ones that are FDA approved included Romidepsin, Belinostat, and there's also an oral form of HDAC inhibitor called Chidamide that's approved in China. And I know that there are many other forms of HDAC inhibitor, including oral agents, are in clinical development. In general, HDAC inhibitor as a single agent has modest activity. They usually have a response rate in 30% range with some complete remission. So using that in combination with other novel agents or in combination with chemotherapy perhaps would improve their response rate and make it more effective in T-cell lymphoma. And we do have examples of that. A recent study that was published that evaluate combination of Romidepsin plus lenalidomide as doublet and Romidepsin plus lenalidomide plus calfezimib as triplet. Those combinations, when you compare that with single agent Romidepsin, they do show increased overall response rate now in the 50% range. And certainly we can envision that more patients would have benefited if they were given combination treatment, as long as the side effects are tolerable. And the other, development trend would be try to move those effective combinations from relapse refractory settings to frontline settings. In doing so, perhaps increasing treatment sensitivity and prevent resistance development at even early treatment stage. And the other therapeutic targets, I think that's receiving a lot of attention for development is PI3 kinase inhibitor. So there are many signaling pathways that are important in T-cell lymphoma pathogenesis. One of them is the T-cell receptor signaling pathway, PI3 kinase, AKT, mTOR. Those are important players. They also interact with cytokine-mediated signaling pathway that going through JAK-STAT. So PI3 kinase inhibitor, for example, JAK inhibitors, they are being actively evaluated for their activity in dedicated T-cell lymphoma studies. Some actually coming with quite impressive clinical efficacies in T-cell subtypes. Examples including duvelisib. It is a delta form of PI3 kinase, and it is currently undergoing pivotal phase 2 trials in relapse refractory T-cell lymphoma. Based on early studies, we're looking at over 50% overall response rate and good CR rate as well as a single agent. And one can envision that this in combination with epigenetic-targeting agents, or this in combination with chemotherapy, and with the hope that they would provide improved synergistic response rate for subtypes of T-cell lymphoma. Lastly, this is certainly a brief review. So the other aspect would be the tumor microenvironment, where immunotherapy perhaps would contribute to disease control and activity for patients with T-cell lymphoma. The earlier compound included immunomodulatory agents such as lenalidomide, but newer agents include immune checkpoint inhibitors such as anti-PD-1s. And I think we are learning to see what are the T-cell lymphoma subtypes that are particularly susceptible for this type of immune manipulation, particularly PD-1 blockade, and try to steer the treatment towards those subtypes. We have also learned that certain subtypes, such as ATLL, patients may have hyperprogression when you subject them to PD-1 blockade. And therefore, those certainly would be, contraindicated for PD-1 type of strategy. And I think that there are certainly other immunotherapy agents that are in very early development, including bispecific antibodies, CAR T cells. And I think challenge and the promise there is that you have to look for very specific T cell antigens where those immunostrategy would target lymphoma-specific cells, but actually leaving the host's immune system intact. So, I think it's very exciting that a lot of new agents are in development on the horizon. Many of them are getting into more advanced clinical development, either as a single agent or in combination, not only in the relapse setting, but start to be moved into the frontline setting, at least in the experimentation phase. And I think all of this would be translating into a good news for our patients. They would have earlier access to novel agents in their treatment journey. And overall, the hope is that, we would come up with good combinations where treatment response would be higher and side effects would be more tolerable. And we would be looking at improved survival, longevity, and as well as preservation of quality of life. So all of those are very important objectives for T-cell lymphoma therapeutic development.